Welcome everyone, and thank you for joining our 2020 Capital Markets Day, which is obviously being held virtually for the first time. We're going to focus today on the process we have for launching the first products. I'll start with going through the agenda, and then we have the vet side. Dr. Wilson Robles will go through the technology itself, and also Dr. Michelle will tell us product rollout going forward as the vet product. And I'm very happy to say today we're on target at the moment for the launch this year. Next, we have Dr. Terrell, who will be telling us about the blood cancers, which have worked so well in dogs, and our plan for launching them in humans. So I'll start with our pandemic focus. So of course, during COVID, we've been very careful to keep our team safe. Our lab in Belgium has not closed for one day. We had a small closure in Texas A&M, and those of us who can work have been working remotely. We've also worked very hard to strengthen our balance sheet and secure the supply of our key components. And I think we looked very carefully strategically on what we can do during COVID, what we can't, and we decided we should really focus on simplicity. So of course, the vet side is a very key one. The COVID side is also very key. And I'd also like to reiterate very strongly, we're still very serious about our marquee lung and colorectal cancer programs in humans. And I'd like to thank yet again our team for really working hard during this time to make sure we stay on track. And I'm very happy to say yet again, we've met our milestone, and the lab, the new facility, Silver 1, is about to open in Namur. Just a quick one on the COVID side. We're really excited with the data we've had so far from the COVID. So there's 3 levels to the trial. The first one was very successful. Can we measure COVID from healthy? There's no point doing this if it doesn't measure COVID. We had 98% success, which was fantastic. The next step is, could we tell different signals from how sick you are? And that worked out extremely well. So now we're just seeing longitudinal samples from the same people to see how it progresses. And we have just finalizing the details of 2 fantastic prospective studies in the US and in Europe. So I'd like to pass it over to the now Professor Wilson Robles, and she will outline the product from the veterinary side and why vets will use it. Heather?
Good morning. This morning I'm really excited to talk to you about some new data that we recently presented this past weekend at the Veterinary Cancer Society meeting regarding the use of the veterinary Volition product, the cancer screening test, in 2 very common cancers that we see in dogs, both lymphoma and hemangiosarcoma. We collected about 134 normal, healthy animals, as well as 73 dogs with hemangiosarcoma and 127 dogs with lymphoma. And now, if you look at our normal levels here, what you can see is the healthy dogs have a very consistent level of nucleosomes. And so that's really tight, really nice. Every dot represents a case. Moving into the data we got from the dogs with lymphoma, I think you can see there is a big difference between these guys. So this graph over here on the left is basically showing you the healthy dogs. You can see how tight the normal values are. They're just right down there at the bottom, all clustered together. However, our lymphoma dogs have a much bigger variety of nucleosome levels in their blood. When we get into the hemangiosarcoma patients, the data gets even better. And so if you look here, what you'll see again, that healthy group of dogs is really tight and low there at the bottom, but our hemangiosarcoma cases are even more spread out, and there's even fewer of them that sit in that lower range that could be confused with a healthy dog. So moving forward, we're pretty excited about being able to use this assay with other cancers too, and then other species as well. So this concludes the scientific portion of the presentation. We are really moving forward, ready to launch this product. And with more information on that, I will pass this on.
So I would like to start this presentation by saying it's amazing because we're starting to talk about a product launch already. So thank you to all the team members who have been doing a phenomenal job. Volition Veterinary has reason to believe that the product will be a great success because we have just shown that in a study of over 330 dogs, and if we put ourselves at 100% specificity, the new QVet cancer test can identify 74% of lymphoma and 89% of hemangiosarcoma compared to the control population. So these numbers are phenomenal. I will now transfer to Dr. Jason Terrell, who's our Chief Medical Officer, and he will now talk to you about the work that we have done in blood cancer in humans. Thank you.
Hi, I'm Dr. Jason Terrell. I'm the Chief Medical Officer for Volition, and I'll be discussing the exciting work we're doing in blood cancers. I'll be reviewing the data we have to date, as well as talking about 2 studies we have planned for the near future. We have looked at 280 blood cancers, and these include non-Hodgkin's lymphoma, the acute myeloid and lymphoid leukemias, compared to 144 healthy individuals. And the clinical question that we wanted to ask is, does our assay distinguish the blood cancers from the healthy population? And so far, we've seen that a single Nu-Q assay, H3.1, produced excellent AUCs or accuracies that range from 85 to 88% for both newly diagnosed and total patients. The single largest and most recent study we've performed looked at 74 cases of newly diagnosed non-Hodgkin's lymphoma versus 72 healthy individuals. And here again, we saw that the single Nu-Q assay, H3.1, detected 61% of the cancers if you set the specificity very high at 95%, and all the way up to 72% of cancers if you back the specificity down to 90%. The first new study looks at non-Hodgkin's lymphoma, making the original diagnosis in pre-treatment patients. And this is a U.S. regulatory trial, meaning it is purposed for FDA approval. The second study will be the first time Volition has looked at treatment response in any cancer. And for this, we're looking at diffuse large B-cell treatment. Well, that concludes the brief talk on blood cancers. Thank you.
Thank you very much, Jason, and thank you very much, Heather and Gatan as well. Our vision of creating an epigenetics platform that can be used in a wide range of areas is absolutely coming to pass now, and I'm very excited that we're on the cusp of launching our first products. We'll have other capital markets days coming up soon, so I'll now pass it over to our analysts for the questions. Thank you very much.